I have the honor and pleasure to give the first session today. I'll be discussing drug-and-drug interactions between small molecule inhibitors (SMIs) with direct-acting oral anticoagulants (DOACs) in patients with lung cancer. These are my disclosures. But my goal now is to take you on a journey so that these conclusions here on the right side of this panel will make sense to you. Assess, avoid, and follow up. And we'll have to get there in 15 minutes, no less. 15 minutes. We'll be able to do it perfectly. So first, I'll focus on cancer and lung cancer specifically. Cancer, as we know, confers an increased thrombotic risk. There's a clear causal link explained by a prothrombotic state. In cancer, each of the three components of Virchow's triad that predispose for thrombosis have some abnormalities. For instance, many coagulant factors such as tissue factor are increased. These mechanisms are likely to be multiple and synergistic. And this prothrombotic state leads to an increased risk of symptomatic VTE, such as deep venous thrombosis and pulmonary embolisms. In lung cancer, this increased thrombotic risk translates into an annual VTE risk of about 14%. This is high, as you can see here in this graph with data from a large claims database analysis from the US. Importantly, we think thrombosis, so we think about anticoagulation, but there might be some issues. There is a reduced protection by anticoagulation. Here we see data from a prospective follow-up study where they found that cancer was associated with an increased risk of recurrent VTE among those who receive anticoagulation. You can see here on the left panel. On the right panel, you can see an increased risk of major bleeding among those with cancer compared to those without cancer. So in sum, and this is the first bit, lung cancer is associated with an increased risk of de novo VTE and an increased thrombotic and major bleeding risk among those who receive anticoagulation in the management of said VTE. For cancer-associated thrombosis, we surely need to give anticoagulation. Anticoagulation is indicated, and DOACs are a very appropriate option. And this is also recommended explicitly in multiple clinical practice guidelines. The latest ESC guidelines correctly state that apixaban, edoxaban, rivaroxaban, all 10A inhibitors receive a 1A indication in this setting if there are no contraindications. And they do draw our attention to so-called major drug-drug interactions where low molecular weight heparins might still be favored over DOACs, over these three selected DOACs. So also here a short summary. DOACs are non-inferior to the standard of care, the low molecular weight heparins. There is a signal of improved thrombotic protection, but there might be a price to pay regarding more bleeds. And we should take into account major drug-and-drug interactions, which is, of course, the main topic of this talk. First, drug-and-drug interactions are so prevalent in the cancer setting that the American Heart Association has put out an excellent review paper on this topic, how to manage these interactions. I highly recommend this paper. It's very nuanced and rather complete, actually. There are, of course, many potential drug-drug interactions in the domain of cardio-oncology, and here we'll focus on those related to NOACs, specifically in lung cancer, where the SMIs might impact DOAC exposure. So the SMI is a culprit, and the DOAC might be a victim drug. Here, I'll be using these 4 steps to give a sort of helicopter view on how to approach drug-drug interactions with SMIs and DOACs in patients with lung cancer. First come the basics. The ADME, absorption, distribution, metabolization, and excretion, absolute basics. And very briefly, you have these 3 DOACs, or NOACs, with data in cancer-associated thrombosis. I'd like to draw your attention here to the right side of the slide. All of them work fast. They work rather short as well, and then you arrive quickly at steady state, and similarly, within 2 days of stopping DOAC, most, if not all, anticoagulant activity will be dissipated, and all of them undergo some degree of renal elimination. In that sense, you can consider them to be rather oral analogs to low molecular weight heparins. They kind of work the same way in our body. And these DOACs have a much lower drug-drug interaction potential compared to vitamin K antagonists, for sure, but it is not zero, particularly if you compare them to low molecular weight heparins. To different degrees, these DOACs are substrates to several enzyme systems, channels, proteins, and here I'll focus on the CYP enzymes and PGPP glycoprotein. But what does this now actually mean? What does this convey in the setting of DOACs? Well, this is actually quite straightforward. Exposure will decrease in the presence of inducers for all three of them. A strong pan inducer is rifampicin. The exposure will increase with the use of strong dual CYP3A4 PGP inhibitors for apixaban, rivaroxaban. Typical dual inhibitor is ketoconazole, and for edoxaban this will occur in the presence of strong PGP inhibitors. A typical inhibitor that might be relevant in this setting is cyclosporin. This is also what we can recognize in the package label of these selected DOACs. It's quite explicit, actually. So here you'll notice already that SMIs are not included in the package labels. And as a rule, the DOAC will not influence the SMI, but the SMI can act as an inducer and/or inhibitor and hence impact DOAC exposure. But how to deal with these SMIs then in this specific setting? Well, it might be interesting to take a look here at guidance from the FDA and EMA on how to interpret the relevance of a given drug and drug interaction. And they consider a drug and drug interaction strong in the case that it confers more than a 5-fold change of the area under the curve, moderate in case of a 2 to 5-fold change, and weak if it's less than 2-fold. For example, here we see a graph of apixaban exposure. On the x-axis, you can see the AUC ratio of apixaban taken together with a specific agent versus apixaban alone. And the one above is ketoconazole, which it doubles the AUC of apixa and is relevant. And this is rather a conservative approach, or a simple, very straightforward, not really nuanced approach. Doubling or halving of exposure is actionable. And again, this is what informs the package label, and we can recognize this. We do have some trial data on what happens when we actually double the dose, and the bleeding risk will go up. It is then increased by up to approximately 1% when you double the dose. Einstein Choice, Amplify Extended were extension trials in the VTE setting, a secondary VTE prevention engaged TMEAF. 48 was the landmark trial of edoxaban in AFib. Okay, so besides this framework, trial data might also be informative. First, we have some good news. Most interactions are not clinically relevant, so that's a good thing. But then, of course, the practical news, we could take a look at the actual trial protocols. SELECT-D with rivaroxaban and Caravaggio with apixaban did not provide real explicit guidance on how to deal with drug interactions. And in Hokusai VTE cancer, well, the protocol did stipulate how to dose edoxaban in case of certain co-medications, as detailed here on the slide. But now, when we take a closer look here at Caravaggio, only selected drugs were excluded. So it's an exhaustive limited list. And as a result, many potentially interacting agents were allowed. And these included potentially interacting cancer therapies. In a post-hoc analysis, the influence of these potentially interacting co-medications was investigated on multiple outcomes. And as you can see here, for recurrent VT on the left, major bleeding on the right. There was no real impact associated with the use of these interactic agents. It's actually quite reassuring. And similar results were retrieved for death from any cause and event-free survival. Also reassuring. So in sum, there was no signal of any harm for any of these endpoints when taking CYP, PGP inhibitors, or inducers together with apixaban in the setting of cancer-associated thrombosis. But, always a but, such analyses are reassuring for sure, but there are some limitations. About 25 had lung cancer in Caravaggio. A little bit more than half of all patients received active chemotherapy, but actually only a very small number of patients received targeted therapies, received small molecules, small molecule inhibitors. So it was limited. So again, while reassuring, we cannot really draw strong conclusions regarding specific therapies. Perhaps plasma monitoring might help us to interpret the impact of these agents on DOAC exposure. And here I've tried to give you the shortest possible summary on how and why of fixed dosing that we follow for DOACs in clinical practice. There is a clear relationship between dose and plasma exposure, at least in phase 1 and 2 studies. And this plasma exposure, again in a linear manner, is linked to pharmacodynamic effects. They went then ahead with fixed dosing phase 3 studies, but other things came into play. Patient characteristics do not only impact the dosing and the exposure, but also outcome. Moreover, posthoc on therapy ranges were retrieved and documented, but they were very broad, and they were based on limited sampling performed as part of said phase 3 studies. And given the clinical results attained with such broad ranges, some argue that with DOACs, we might have ended up with a sweet spot. And taken all of the above together, I propose to you now a pragmatic framework. So we have seen that DOACs behave like low molecular weight heparins with a high propensity for drug-and-drug interactions, depending on the DOAC. Most therapies do not lead to a doubling or halving of steady-state AUC and are actually allowed. Clinical data up to now are rather reassuring, and plasma monitoring might have a place, but we should refrain from over-interpreting these data. So what would now be a feasible, pragmatic approach? And here on the left, you can see the proposal on how to think about, how to assess, how to select anticoagulation in the setting of cancer-associated thrombosis as recommended by the European Society of Cardiology. On the right, I've added some very basic steps to help professionals. And step 1 is think and assess, consult with an expert. Please use good software prescribing software and consult valid databases. Step 2, well, we should be reassured that there are many drug-drug interactions and most are not that relevant. We could draw inspiration from the FDA EMA framework. For example, if the impact is less than 2-fold, most likely it's not that relevant, and otherwise it is actionable. You should do something, and here I'd argue to select an alternative, for example, another DOAC with fewer drug-drug interactions or a low molecular weight heparin. And then step 3, try to keep using these tested doses, refrain from adjusting doses based on solely pharmacokinetic data. We can and sometimes should probably measure exposure, but mostly to exclude over and under exposure. And of course, therapy should be evaluated periodically. Regarding those databases, I'd like to add that all published work is easily dated. For example, you can see here the table from the ESC guideline, not interactive, no background literature, difficult to use. What then to use? Well, you have multiple options, for example, the DDI manager as seen here. I've added some additional nuance, but this is just additional information. Most importantly, also take into account the limited time, I'd like to go to the take-home messages, particularly now that all of us can understand these 3 main conclusion. We should assess and consult. We should avoid situations where the exposure is doubled or halved. Follow-up should be provided, and if need be, we can measure to exclude over or under exposure. I thank you for your attention.